Hello everyone, this is Rohan Meshram, and today is our third session on my lecture series on protein structure visualization and analysis. And in today's session, we would be dealing with some basic exercises in protein structure visualization. Okay, and the main objective of today's session is to just demonstrate you on as to what are the different ways of selecting and displaying the different amino acid residues that are present in your protein, and what interesting things can you infer about a protein's structure and eventually the protein's physiology by simply selecting and displaying your protein molecule in different orientation. But before we do that, I would like to spend some time discussing some very basic aspect of protein physiology. Okay, so now, the main objective of this doing these practicals and doing this structure related practical of protein is that proteins are very important in cellular physiology. Okay, so as you people might be already aware that each and every aspect of cellular physiology is either directly or indirectly connected to protein. Each and every function that is needed to be carried out in your cell in order to keep it alive, well, all those uh, all those functions or all those activities are governed by protein. They are carried out by protein, or at least they are influenced by protein. So proteins are one of the most important biological macromolecules that enables life to uh, function properly, that eventually enables life form to perform their activities and to complete their life cycles. So without protein it is not possible to imagine life on earth. Okay, moreover, I would just like to give some statistics. Okay, so as to why proteins are important and why it is important to learn about its structure, why visualization of protein structure is so damn important. Okay, so the thing is that now if we agree on the fact that without protein there would be no life on earth, because most of the cellular physiology is now completely dominated by proteins activity. It can be in form of proteins can act as an enzyme, they can act as messenger, they can act as the structural component that sustain the integrity of your cells. Okay, so they work in each and every aspect of cellular physiology. Okay, so basically these proteins are of variety. Proteins have to perform a variety of function. Okay, I would just give you some facts by which you can just imagine what might be the diversity in functionality that this protein has to achieve. Okay, well, say for in our case of humans, we know that there are some 40,000 genes that are already known to exist. Okay, so there are some 40,000 protein coding genes that are known to be present in human. Okay, and this is only about human. Okay, so there must be different type of proteins in different type of organisms. Okay, and life is full of diversities. We have known some bacterial or say some sort of organism that sustains in highly acidic condition, or there are some life forms that exist in highly extreme conditions, you can say, whether it can be in form of sustaining very high temperature or sustaining very low temperature. And life has kind of evolved to withstand any environmental pressure, and such withstanding mechanism is usually dealt with some unique proteins that are expressed in each and every organism. Okay, so now you can imagine that how many organisms are there in our biota, right? There are billions and billions. Some we know, and some of them we haven't even discovered, right? And each and every organism in unique, they have got their own set of proteins that they express at different times in their life cycle. Okay, and in this way there are millions and billions of functionalities that these proteins are interested to be to be performed in order to sustain life on earth. So now, strictly speaking, chemically, your let me write this thing. Okay, so there are billions of functions that are carried out by proteins. Okay, now second fact that you should learn, if we talk strictly about the biochemical or chemical nature of protein. Proteins are, you can say, are nothing but polymer of amino acid residues. Okay, the polymer of amino acid residue, you can say that there are 20 standard amino acids that constitute the proteins as such. And these 20 standard amino acids are they are attached to each other by peptide linkage. Okay, so we'll say, for example, you can represent any protein. We usually represent the protein's structure starting at the N terminus. You have got this NH2 at N terminus, then you have got C alpha carbon atom. The C alpha carbon is attached to its side chain. Okay, then we have got carbonyl carbon atom, then we have got an oxygen that is attached to your carbonyl carbon CO group. Then you have got this nitrogen. Okay, and this nitrogen is again attached to another C alpha carbon atom. Okay, you might have some another side chain group attached to it. Again you have got carbonyl carbon, again you have got nitrogen. Okay, and this go on continuing. Okay, so this particular bond CONH, it is called as peptide bond. Okay, and this is the first amino acid residue, this is the second amino acid residue, so on and so forth, it would continue. Now the side chain of each and every amino acid residue would differ, and depending on what is the side chain of that amino acid residue, each and every amino acid residue has got some interesting and different physiological properties. Okay, so as I have already said that there are 20 standard amino acid residues. Okay, so I would just list out those 20 standard amino acid residues. Okay, they are in letter like A, you go A, then you have got C, D, A, that stand for alanine, C, that stand for cysteine. Then you have got D, E, F, G, H, I, J, K, L, M, N, O, P, Q, R, S, T, U, V, W, Y, Z. We don't have this. O. This A stand for alanine, C stand for cysteine, D for aspartate, E for glutamate, F for phenylalanine, G for glycine, H for histidine, I for isoleucine. We don't have anything for J, K for lysine, L for leucine, M for methionine, N for N stand for asparagine, P stand for proline, Q stand for glutamine, R stand for arginine, S stand for serine, T stand for threonine, V stand for valine, W stand for W stand for tryptophan, and Y stand for tyrosine. We don't have Z. So these are all 20 standard amino acid from which at least protein from planet Earth are made up from. Well, now I would just like to kind of raise a question. Like, we have come to a kind of consensus that there are billions and billions of functions that are needed to be carried out by proteins, and these proteins are nothing but polypeptides, or you can say polymers of different amino acid residues, and we have got only these 20 standard amino acid residues. Okay, so now one might ask a question as to how is that only the combination of these 20 standard amino acid residue would yield such a functional diversity in your protein. How is it possible that these proteins, which are made up of only 20 standard amino acid residues, are able to perform such functionally diverse activities? Right, so that might be a daunting question, right? So my friends, to answer this question, well, you should now try to learn that these proteins do not remain linear as you might have imagined so far. So these standard polypeptide chains of these proteins, well, they are not always linear, right? The protein eventually folds. Okay, it folds upon each other. The structure of protein is such a way that it acquires a specific three-dimensional structure, and it is its three-dimensional structure that enables our protein to perform its function. So it is not just a series of linearly arranged amino acid residues that are joined by peptide bond that would enable your protein to function properly, right? So in order to perform its function, your protein must fold, right? Protein folding is very, very important in order to perform its function. If the protein is not folded properly, it will not perform its function. It would be unable to carry out its physiological role if it is not folded properly, in or if it do not take a specific conformational state that is needed for its activity. So this is what it is. It is the sequence of your protein which enable it to fold properly, and if it is folded properly, it would perform its function. So this sequence to structure to function is holy grail of structural bioinformatics, right? In order to perform its function, it is important that it has some certain three-dimensional structure, and the three-dimensional structure of your protein would eventually determine by its sequence. Now it is completely a different story as to how proteins know how to fold. Okay, so that would be a completely different aspect of protein physiology that is way out of scope of our discussion for time being, right? So for time being, if you understand that it is the primary sequence. What is the primary sequence? Sequence means what? The order in which the amino acids residues are arranged in your linear polypeptide, we call it as the sequence. Okay, and this sequence is known to fold in three dimension. It is known to acquire certain specific three-dimensional state, and that three-dimensional state would allow your protein to function. Okay, and this step can be called as the holy grail of structural bioinformatics, right? So now it comes now our discussion has come to a point for you to you know let you it is now for you to understand that unless and until your protein folds properly, it cannot function. And for folding, it is the sequence is the sequence would eventually guide the protein to fold, and after folding it is able to perform its function. This is very important for you to learn. Well, now protein is said to be folded at different level, or you can say the protein exists at different structural level. What we call it as the primary structure of protein. Okay, so the primary structure of protein is nothing but the sequence of amino acid residues. Then the primary structure, the next structural level at which the protein fold is secondary structural level, wherein the primary structure folds into small local regular structures. Well, say for example, the different type of secondary structural units that are existing in protein are in form of helices, strands, turns, coils, or you can also call it as loops. Okay, and eventually you have got tertiary structural level, in which these secondary structural elements would fold upon each other to give some three-dimensional state of your protein. Okay, so these helices can be attached by loop, and there can be secondary structures like these are beta strands, and this beta strand would form beta sheets. These are helices, these are loops. Okay, so the fact is that when these secondary structural elements fold upon each other in three dimension, they would form this tertiary structural level. Okay, there is another level called as quaternary structural level. Okay, if your protein contains multiple chain, then these multiple chain in their tertiary structural level can come together to form quaternary structural level. Okay, so this is, you can say, the different levels at which the proteins can fold. Okay, So now in biochemistry point of view, or in medicinal chemistry point of view, or in bioinformatics point of view, why is that I would be interested in learning this different structural level or different protein structures as such? Why should I learn about these protein structures at the very first place? Okay, well, most of the well, this is for the medicinal chemist people. Well, most of the uh, drug targets, these drug targets are usually enzymes. They can be channels, okay, or they can be receptors that are present in cells, and these all of these are eventually protein in nature. So it is, and these enzymes, channels, and receptor eventually are main drug target. Okay. So since these enzymes, channels, and proteins, channels, it can be ion channels, it can be receptors like G protein, enzymes like DHFR, cyclooxygenases. Well, all these drug targets are eventually proteins in nature. So unless and until you understand what those enzymes or what those ion channels or how those receptors function, how those receptors look like, you won't be able to synthesize or create effective drug target. So it becomes very, very important for you to know what is the structure of your drug targets in order to design some appropriate drug molecules or inhibitor molecules that might have certain therapeutic significance. Okay, so that is why it is very important for you to learn about protein structures. Okay, so now said this, I hope you people now agree with me that it is the protein's structure plays a very important role in its physiology, not only in context of its application as therapeutic drug targets, but also in sustaining and maintaining life on earth. So that is why learning about the structures of given protein is very, very important. And this is the exact reason that I have designed this lecture series, so as to enable uh, you to have an insight on as to how proteins are, and what can be manipulated, how it can be manipulated, where it should be manipulated. Okay, so well, say for example, if you are good at analyzing protein structures, it would become very easier for you to locate as to where is the active site of protein, where is exactly that my drug would go and attach to that protein. So all these facts would become very easy for you if you are well versed with analyzing protein structures. Okay, so now let us begin with our practicals or demonstration on protein structure visualization and protein structure analysis. Okay, so well, in most of our practical session or demonstration session, I would be using the software called as SPDBV. Okay, it is also sometimes called as deep view, right? SPDBV stand for Swiss PDB viewer, or it is also sometimes referred to as deep view. Okay, so now there are other options available. Okay, people might use Chimera, they can use VMD for doing the same stuff. If you are working at very elementary level, even RASMOL, is also very handy, but I have selected this deep view for most of the practical, because it is quite visual. You don't have to remember any kind of commands in order to visualize your protein molecule or perform structural analysis. It is all completely dependent on a graphical user interface, so it becomes easier for you as a beginner to start out with such a program. Okay, once you are well versed with SPDBV and you know as to how to manipulate the protein structure and how to visualize, then you can use advanced tools like VMD and Chimera, which has got certain more added advantage over visualization. Okay, but they also require some scripting or say some programming language in order to use it properly. So for time being and for the sake of simplicity, we would be using our Swiss PDB viewer for our subsequent practical session or demonstration sessions. Okay, so well, let us now begin with our practicals, right? So well, today, specifically today's session, we would be I would be dealing with uh, very basic exercise. Like, I would show you how do we select and display the different parts of your proteins. Okay, so when you begin with when you begin your career in protein modeling or molecular modeling, well, it is advised that you should at least have command over any one at least of the protein visualization software. That would make your life so easier. Okay, so I have selected to train you on SPDBV. You can also start out with either Chimera or VMD as well. Well, these softwares are also very good. Okay, so at the end of this semester or at the end of your course, you should be at least well versed with one molecular modeling package. You should be able to develop your own style of analyzing the proteins. Okay, that is what my objective is. Okay, so in our previous lecture, I have told you as to how do we download it and how do we install it. Okay, I have just double click on the icon on my desktop in order to open our Swiss PDB viewer main window. Okay, and I have already downloaded our protein molecule called as 1HEW. So what we will do is that we will start learning by looking at the enzyme lysozyme. If you remember from my previous lecture, that 1HEW is lysozyme from hen egg white. Okay, so we would start out by looking at the enzyme lysozyme that is in complex with this trisaccharide inhibitor. We would further refer to this inhibitor as trineg. Okay, and the atomic coordinate of this model are contained in protein data bank file with code 1HEW. So I have also demonstrated you in our previous lecture as to how do we download this protein molecule from PDB website as such. Okay, so to begin with, uh, the very first objective of our today's demonstration is to teach you how do we select and display molecule in different way. Okay, so when you load your molecule, well, you can well, let us start out with this control panel. Okay, so now you can open this control panel from this Windows option. Go to Windows and click on this control panel. You would open this control panel. Okay, so the control panel is used to select or display or hide the part of the model. Okay, so in your deep view user guide, the parts of the structural model of your protein, well, they are called as groups. Okay, these are called as groups, and these groups eventually mean the amino acid residues or whatever hetero groups in terms of inhibitors or prosthetic group that are present in your model. Okay, so this control panel would eventually list out the amino acid residues and the inhibitor or prosthetic groups that are present in your protein molecule. Okay, so this particular protein, say for example, uh, well, is made up of amino acid residues, okay, the protein molecule, as well as your inhibitor that is co-crystallized with it. Okay, so now let us talk about how do we select the group. Okay, so selecting the groups or selecting the amino acid residues. Okay, so now selecting the amino acid residue will not change what it is shown on the display menu. Okay, or what we call it as the graphical window. Okay, but whatever you select, but the actions that are taken on the model would affect only the currently selected groups. Okay, that is the difference. When you kind of select the amino acid residues in your control group, it won't change anything over here. But if you take some action, that action would be carried out on only the selected group. Okay, so say for example, your control panel lists all the groups in the model, and this control panel thus provides a fast and very powerful means to select, hide, and display various amino acid residues that are present in your protein. Okay, so what we would do first is that I can simply scroll down to the bottom of the list to see how many amino acid residues are there in our protein, right? So I have just scrolled down. You can use this scroll bar to scroll down and see key will say how the hetero groups are arranged at the end of your PDB file. Okay, and hetero groups are usually conventionally placed at the end of the list as such. Okay, so hetero groups are usually present at the bottom of the amino acid residue list or the participant list in your protein structure. Okay, so now the trinag inhibitor is listed as 3 residue of NAG. NAG stand for N-acetyl glucosaminic residues. Okay, so this trinag inhibitor, this is your trinag inhibitor, is listed as the 3 NAG residues that are numbered as 201, 202 and 203, 201 to 203. Okay, so now let me scroll back upwards. Okay, so now let us I will show you some tricks and tips on as to how you can select various residues by various means. There are different ways of selecting and displaying the residues. Okay, so now what you can do is that you can just click and drag. Okay, well, say for example, I would click and drag starting on the word glycine 4 at the top of the window. Okay, we have reached at the top of the window, and you can, once you have clicked it, you can just drag it down till say for example, I would select glycine 16. Okay, simply click it and drag it. In this way you have kind of selected the group of amino acid residues starting from glycine 4 to glycine 16. Okay, and the moment you release the mouse button, okay, the moment you release the mouse button, the selection would stop. So I would click by left mouse button and drag it till the residue that I wish to select, and then release my left mouse button. So this is how you can select some specific range of amino acid residue. Okay, so now as you might have noted, all the names of the amino acid residue from glycine 4 to glycine 16, they have turned red. Okay, so now the groups that are shown in red are now selected. Okay, so this is the simplest way to select a small number of residue, is to click at their name and drag to select the range of them. Just click and drag a range of amino acid residue that you want to select. Okay, now if I hit enter button on my keyboard, let us see what happens. Okay, so after selecting, when I hit enter on my keyboard, this action hide all the groups except those are selected. Well, now I have already told you, selection won't make any difference in your display, but whatever action that you take, that would eventually be affecting only the selected groups. Okay, so what action we have taken? We have selected this range of amino acid residues, and we have just hit the enter button. So this would mean that we want to visualize only the amino acid residues that are selected. Okay, so hitting an enter after selection hides all the groups except those that are selected. Okay, then there is a button called as this zoom center button. This button is called as zoom center button on the toolbar. So if you now select this zoom center button, well, you know, this particular whatever the part of the protein that you have selected and display would be centered upon. There is one more thing that you should know. Now if you try to rotate your selected group, now you might note that this it is rotating still around the center of the entire protein molecule. Okay, just note that. We will come to that point in a little while. Okay, so now if you press the zoom center button, now your whatever is shown on display, well, it is being zoomed on. Okay, so now notice that after centering, the model rotates about the center of groups on display. So initially, before pressing the zoom center button, this particular displayed group of amino acid residues were rotating around the axis of the entire molecule. But now, after pressing this zoom center button, it is now rotating around the axis of around the center of whatever has been selected. Well, this makes a lot of difference. Okay, so now what you are noticing is that after centering, the model rotates about the center of groups that is on display. Now what you are doing is that you are looking at the residue 4 through 16 of the protein. Now, do you recognize this type of secondary structure? Okay, do you recognize, or are you able to figure out as to what might be the secondary structure of this protein? You might also notice that there are check marks that appears in the show column next to what has been selected. Okay, okay, and this V, or what is called as the show mark, that stands for visualize. Okay, indicate that only these residues are on display. Okay, so now there are also check marks on the side chain. Okay, this is called as the side column, as you can see over here. Okay, there are check mark also in the side column, which means that side chain are also on display. Okay, but although you can see that there are check marks in front of other amino acid residues as well. Okay, but these side chains are not visible. Why? Because a side chain is only shown if the rest of the residue is shown. So unless and until you show the residue, you cannot show the side chain. Okay, a side chain is shown only if the rest of the residue is shown. So you only see the side chain of displayed residue in the graphic windows. So whatever it has been selected, whatever we have shown, only the side chain residue of side chain of only those residues are visible. Okay, so now let us see what are these different columns that are present over there. Okay, now let us click the check in the side column next to methionine 12. Okay, over here, this is methionine 12. We have clicked on this side column in front of this methionine 12. Do you see what happens? The side chain of this methionine 12, well, it appears and then disappears. Okay, do you see these things? In this way you can manipulate as to what side chains are to be shown and what side chains can be hidden in your protein molecule. Okay, so now let us try out on clicking on the visualization button on the remaining columns as well. Okay, we would try the same thing in this show label, in this show label column, as well as in this surface column. Okay, now what do you see? So this surface column is nothing but a little circle of dots that is present over here. So this is called as show surface column. Okay, and let us see how our display has changed. Okay, so the surface that is now being showed only on methionine 12, it is displayed as a dotted van der Waals surface. Okay, for the group. Okay, so now what you can do is that you can remove these checks from the label and surface column. If you remove this label, the label would be hidden. If you remove this check mark from over here, the surface, the van der Waals surface, will also be removed. Okay, so in this way you can selectively tell as to what is needed to be labeled or in what is needed to be shown in surface representation. Okay, so now we have removed the checks from the label and surface column. Okay, now what you can do is that you can click on this heading from this control panel. Okay, if you click on this heading label, let us see what happens. Okay, what happened? Clicking on this word label at the top of the control panel, it would label all the residues that are selected. Okay, since we have selected glycine 4 to glycine 16, all the residues that are selected are now labeled with a single click. Okay, similarly, if you click on the heading surface, as I am doing right now, okay, the surface is the symbol in a little cluster of dots as you have seen over here. So clicking on the surface symbol at the top of your control panel, okay, would show a dotted surface on all of the residues that has been selected. Okay, similarly, if you click on this button, on this ribbon, heading on this ribbon, it would now show that that secondary structures over here in ribbon form. Okay, so now clicking the word ribbon draws a very smooth stranded cartoon of the selected backbone. Okay, so the clicking a word heading, okay, clicking a word or what we call it as the heading at the top of the column adds the corresponding display for the selected amino acid residues that are shown in red. Okay, so this is how now you can clearly see what kind of secondary structural element this can be. Okay, so you can clearly see that whatever we have selected, well, it is an alpha helix. This is called as alpha helix. It is one of the important secondary structural elements that stabilizes the protein structures. Okay, now let us remove the label, surface, and ribbons. Okay, so now what you can do is that there is a trick by which you can remove the labels or surface or ribbons. What you are supposed to do is that you are supposed to hold down the shift key on your keyboard, hold down the shift key and click any check mark in the label column. Okay, hold down the shift key and click in any of the check mark that are present in your label column. Let us see what happens. Shift and then click. Shift clicking would uh, shift clicking would enable you to carry on certain function on the selected amino acid residue. Okay, you can repeat the same shift click in this surface column, in this ribbon column as well. Okay, so when you click on the check mark while holding down the shift, your action removes all check marks and the deep view makes the corresponding changes in the display. Okay, so that is also one of the trick to visualize all the selected residues. Just shift and click in any of the selected residues. That would eventually that function would eventually act on all of the selected amino acid residues. Okay. On the other hand, when you shift click in empty space, deep view would place check marks in that column besides all the group and take appropriate action. Okay, well, let us try this out by shift clicking several times in the show column. Okay, so here is the show column. Shift click. Okay, it has shown you the entire model, and if I again do the same thing, shift and click in any of the shown column, it would bring or it would just display, or it would hide the display as such. Okay, so now uh, this displays, this shift clicking in show column would display and hide the entire model, right? This would display and hide the whole model. Now with all group shown, we would try to shift click several times in the side column to display and hide the side chain. We can do the same thing. Shift click to show and hide the entire side chains of your model. Okay, shift click, shift click. That is, now you are kind of toggling between showing the side chains of the entire model as such. Okay, so this is how shift click works. Okay, so now let us uh, let us end with side chains hidden. Shift and click. So now you are visualizing the model, only the backbone of your model. Okay, now shift click in ribbon. Let us try to shift click in ribbon to display and hide the ribbon cartoon for the whole model. Just shift click, shift click. Okay, so when you have this, you can clearly see how the protein has folded, right? So now let us end with ribbon hidden. Okay, so now let us end with model showing backbone of residue 4 through 15. Well, let us show only the backbone of residue 4 through 14. Okay, 4 through, sorry, 16, and selecting them, everything else hidden. Okay, now there is one more trick that I would like to share with you people. So the next task I would like to show is that there is difference between selecting and showing. Okay, so now what our next task is, that we would select and show residue 4 through 16, okay, with their side chains, and add residue 17 through 22 as follows. I would show you. Okay, so now click in, what we would do is that now we would click in the show column next to leucine 17 show column. We would click over here and drag down till 22. Okay, do you note what I have done? I have not clicked on the name of the residue. Instead, I have clicked in front of this show column and drag till arginine 21, or we'll say for example glycine 22. Okay, so what we have done, uh, uh, well, this visualization marks, now it has appeared in the show column next to residue 17 till 22. Okay, but these residue names are not turned to red. Okay, so now if you drag it too far, well, say for example, it might happen that that you might have drag too far. Well, our objective was to only drag till 22, and by mistake if you have dragged too far till tryptophan 28. Okay, so what you can do is that you can simply individually click on the check marks and make your view as you want. Okay, so you can do one at a time as well, right? So now we would add check marks in a side column for residue 17 to 22 in the same way. Well, let us add the check marks from residue leucine 17 to glycine 22. Okay, now what do you see over here is quite simple. Okay, now residue 4 through 16 are selected and shown. Okay, from residue 4 till residue 16 are selected and they are on display, while residue 17 till residue 22 are shown but they are not selected. Do you get the difference? Residue 4 till residue 16 are visible as well as they are selected, but residue 17 to residue 22 are on display. They are visible but they are not selected. So let me make this fact very clear, that it is not necessary that whatever is displayed on your visualization window is necessarily selected or vice versa. Okay, so this is the this is the fact that I want you people to learn. Okay, for easily selecting a range of residue, you can just click on the name to select the first residue, then hold down the shift button while clicking in the last residue in the desired range. Okay, so well, say for example, there is another way of selecting the residue. Okay, uh, so if you want to select a small group of residues, you can just click it and drag it till the range that you are interested in. But if you want to select a very large range, there is another way around. Okay, for well, say for example, if you want to select residue from glycine 4 till well, say for example, till some residue which is present at position 45. Okay, then what you can do is that you can just select at the very first residue that you are interested in, click on the shift button and go till the 41st residue that you are interested in. If you keeping that shift button pressed, if you select the next residue, then all of the amino acid residue that were present between the two selected or two clicked clicked amino acid residues would be selected. I would tell you once more. If you want to select amino acid residue for a very large range, then what you are supposed to do, click on the first amino acid residue of your interest, then press your shift button, locate what is the last residue in your selection range, and just click on the last residue holding your shift button. Okay, in this way you can select a big range of amino acid residues in your protein structure. Okay, so we have tried this one. Okay, so now there are also two narrow columns. We are talking about these two narrow columns at the left of the group columns. Okay, so the first column is populated with letter A, as you can see over here. Okay, well, in previous revisions of this entry, this column used to be blank. Okay, यहाँ पर A या B नहीं रह सकता था. Okay, when the current model contains only one protein chain, it might happen that this column may remain blank. Okay, so this represent the chain of your amino acid, chain of your protein. And if there is a single chain in your protein, sometimes it happens that this column is left blank. Okay, so over here we have A, that indicate that we are working on chain A of the given protein. Okay, so if the model contain more than one chain, one letter chain designation would appear in this column. Okay, since there is a single chain, there would be letter A right from the first amino acid residue till the end of your protein. Okay, now if you click anywhere on the chain, or you can say on the letter A or the chain ID of your protein, you can select the entire chain. Okay, if you click anywhere in the first column, what you have done is that you have selected the entire chain. In this case, the entire model, because our model contains only a single chain. Okay, so now if you select the entire chain and pressed enter on your keyboard and pressed the zoom center button, what you can see, you have now shown the entire model, selected the entire model and displayed the entire model and centered the entire model. Okay, so these are the different ways in which you can select different amino acid residues. Okay, so now this deep view select menu is another way round to select different properties of your protein molecule. Okay, so this deep view select menu, provides other commands for selecting different amino acids or different groups of your proteins or different amino acid residues of your protein. Okay, so now what I will do, I would go to this select menu and use this option select all. Okay, and hit enter. Okay, then I would select on this heading side chain. Okay, so the menu command select all would select all of the groups that are present in your protein. After hitting the enter, it would display whatever it has selected, and if you click on the heading side chain, it would add all the side chains to the groups that are already selected. Okay, so now what you can do is that you can go to this tools menu and use this option compute hydrogen bonds. Okay, now things have changed. In a blink of an eye, deep view or your SPDBV viewer finds and display all the potential hydrogen bonds in the molecule, including those involving the side chain as well as those that are involving hetero groups or the inhibitor. Okay, deep view displays hydrogen bonds as green dashed lines. Okay, we will come to this feature in a while. Okay, so for time being, I just wanted to show you there is hydrogen bond counting and displaying tool present in this tool button. Okay, we would come to this in a while. Okay, so now what we have done is that we have displayed the entire model with side chains, with everything is selected, and we have displayed the hydrogen bonds, so all possible hydrogen bonds in our model. Okay, now let us do something very important. Okay, now let us go to this select menu. Okay, and let us select the secondary structure, and we would select only helices. Okay, now you can see that only some specific group of amino acid residues in your control panel are selected. Okay, and let us push enter in our on our keyboard. Okay, now you should see that only the alpha helical region of your protein lysozyme with hydrogen bonds are shown. Okay, you can see only the alpha helices that we have selected are on display. All the other secondary structural elements are now taken off from the display. Okay, now let us turn off all the side chains. Okay, you can do it by any way. Okay, so I would shift click and let go of all the side chains. So now you can see that there are only backbones of all of the alpha helices that are present in our protein with the potential hydrogen bonds. Okay, so now what you are seeing is that you should see only the alpha helical region of the lysozyme with hydrogen bonds that are shown. Okay, after we turn off the side chain, well, let us look around to confirm that the segments shown are all helical. Okay, these are all helical, right? So now let us focus on any one of the alpha helix. Let us check out with the biggest alpha helix, this one. Okay, let us focus on this and let us zoom center it. Okay, so now I would want to show you one very important structural property, or I would wish you to learn very important structural property. For this we would need to label the amino acid residues. Okay, so now if you note that the hydrogen bond of the first residue is usually with the fifth residue. Okay, so well, say for example, well, let us turn around. Arginine, the hydrogen bond of backbone of arginine is with you know the subsequent 5 residue apart. That is, arginine at 5th position is hydrogen bonded to alanine at 9th position. Similarly, cysteine at 6th position is hydrogen bonded to alanine at 10th position. Subsequently, glutamate at 7th position is hydrogen bonded to alanine at 11th position. Okay, so now can you note some pattern that such type of hydrogen bonding is termed as n to n plus 4th type of hydrogen bond. Okay, that is we have now confirmed that most of the hydrogen bonds are formed between the carbonyl oxygen of residue n to the amide nitrogen of residue n plus 4. That is, the carbonyl group of nth residue forms hydrogen bond with the nitrogen atom of of n plus 4th amino acid residue. Okay, so now depending on what is the type of hydrogen bonding, there are different classes of alpha helices. Well, what I have shown you is n to n plus 4 type of hydrogen bonding. Okay, what I have shown you is simply n to n plus 4th. That is, first amino acid residue would hydrogen bond with fifth amino acid residue. Second amino acid residue would hydrogen bond with sixth. Okay, so on and so forth. So this is called as n to n plus 4 type of helix. Similarly, there are n to n plus 3 type of alpha helix, where there is hydrogen bonding between first and fourth amino acid residue instead of first and fifth amino acid residue. Okay, now depending on what type of hydrogen bonding exists in your proteins, there are different type of helices that you can classify. Okay, so this is one thing that is important. Another thing that you should note, that hydrogen bonding that is observed in this alpha helix or alpha helical as secondary structure is always within the amino acid residue that are present in the same helix. Well, remember this fact. By that what I mean is, hydrogen bonding that is present in your alpha helices are always intra. Okay, that is, one residue from alpha helix would form hydrogen bond with the residue that is present in the same alpha helix. Okay, so that is what we can say, the hydrogen bonding in alpha helices intra hydrogen bonding pattern. There is intra-hydrogen bonding pattern in case of alpha helices. Okay, we'll come to this point a little bit. Okay, so now what have you learned about the secondary structure? It is the type of hydrogen bonding that is present in secondary structure, based on which you can classify those alpha helices. The alpha helices can be n to n plus 3 type of helices. This can be n to n plus 4 type of alpha helices, so on and so forth. And second important thing that we have learned about alpha helices is that the hydrogen bonding pattern is always intra. You have got intra hydrogen bonding pattern in alpha helices. Okay, now let us compare what happens when we select and display another type of secondary structural element. Okay, okay, so now let us go to this select menu, go to this secondary structure. Okay, and instead of helices, now would select strands. Okay, now you can see entirely different set of secondary structural elements are selected. Okay, so what we have done, we went to this select, then secondary structure, where is secondary structure, and then we have selected strands. Okay, when you hit enter and again press zoom center, now you can see that instead of helices we are now displaying the beta strands. Okay, now what you are actually saying is that you can see only strands of the beta pleated sheet. So now you might notice the characteristic pattern of hydrogen bonding in strands. Okay, so as I have already mentioned you, that in case of alpha helices, okay, let me show an alpha helix over here. In case of alpha helices, the hydrogen bonding was intra, right? But in case of beta strands, the hydrogen bonding is inter. That is, one residue from one strand would form hydrogen bond with another residue which is present in some another strand, right? And therefore we can say that inter type of hydrogen bonding pattern stabilizes your beta sheet. So this is called as beta strand, and beta strand collectively, two or three beta strands collectively forms these beta sheets. So you can over here now say that there are 3 beta strands in your beta sheet. Okay, these 3 strands constitute a single beta sheet. So this beta sheet is formed by 3 strands. Over here you have got 2 strands in this beta sheet. So your protein eventually contains 2 beta sheets. First beta sheet which contains 3 strands, another beta sheet which contain only 2 strands. Okay, so now if you click on this ribbon heading, you can see that there are arrows over here. Okay, so this arrow would tell you the directionality of your strand. Okay, so based on how the strands in your beta sheets are arranged, you can have different type of beta sheets now. Okay, if all of the strands in your beta sheets are oriented in the same direction, you can say that beta sheet as parallel beta sheet. You can classify this beta sheet as parallel beta sheet. On the other hand, if the directionality of strands in your beta sheet is alternative, you can call it as anti-parallel beta sheet. And if you have, mixed type of orientation in your beta sheet, you can call it as mixed beta sheet. Okay, so now depending on what is the orientation of your strands in your beta sheet, you can classify them as parallel beta sheet, anti-parallel beta sheet, and mixed type of beta sheet. Well, now let us see what type of beta sheets are present in our protein molecule. Okay, so you can just check out the bigger beta sheet which contains the 3 strand. Okay, one strand goes in this direction, another strand is oriented in opposite direction, and the subsequent strand is again oriented in opposite direction. So it is clearly anti-parallel beta sheet. Okay, this was about the bigger beta sheet, and we have got another smaller beta sheet which is composed of only 2 strands, and this also appears to be having the strands which are oriented in opposite direction. So we have got 2 beta sheets that are of anti-parallel type. That is what you can infer from your protein. Okay, so in this way you can classify these secondary structural elements, okay, as parallel beta sheet, anti-parallel beta sheet. You can also classify the alpha helices based on how the hydrogen bonding pattern is. You can have n to n plus 3 type of helices, you can have n to n plus 4 type of helices. Okay, so now what you can do, you can go to again display, and if you again click on show hydrogen bond, what would happen? Whatever hydrogen bond that you have initially shown, they would go away. Okay, you will find that this command of again going to display and show hydrogen bond, what you have done is that this command has turned turn off your hydrogen bonding. Okay, or you have removed the hydrogen bond from the display. Okay, so you should also notice the other select command menu. Okay, there are also other select command menu. Okay, you can select and display only residues whose side chain carry positive charge. Okay, you can do it by property. Okay, you can select and display only the residues whose side chain carry positive charge, so called as the basic residue. Okay, you can select and display you can select and display only the histidine residue and their side chain. You would have to select by group kind. Okay, group kind would allow you to know as to how many histidines are there or how many tryptophans are there in your protein molecule. Okay, so you just need to check around with that. Okay, uh, so now how many, well, let us see how many histidines are present in lysozyme. Okay, so what we have done, uh, we went to this select, then group kind, and then we have selected histidine. Okay, so now only histidines are selected. As you can see, histidines would only turn to red. Okay, now how many histidines are there? You can check it out using this windows and layer info windows. Okay, you see that over here it would show you how many groups are selected. Okay, so there is a single histidine in your protein molecule. So in this way you can kind of select group kind, and let us see how many aspartates are there. Now you can see that there are 7 aspartate. Okay, let us see, select group kind, how many tryptophans are there. There are 6 tryptophans. In this way you can get an idea as to how many special type of amino acid residues are there, and how this can be handy. I would just demonstrate you with this thing. So in this way, this layer info windows provides the information about what it is on the display. Okay, okay. You should note that this select heading at the top of far right column, this one I am talking about, this column shows the number of groups that are properly selected. Okay, what we can do one more important thing now. Well, let us try and see how many basic amino acid residues are there. Okay, so how would we do that? To select how many basic amino acid residue, we would go to select, group properties, and select how many basic amino acid residues are there. Yes, well, let us note it down. How many basic amino acid residues are there? There are 18 basic amino acid residues. Let me note it down. Basic amino acid residues are 18 in number. Okay, now let us try to check how many acidic amino acid residues are there. Select group kind, sorry, group property, and select. Let us see how many acidic amino acid residue are. So we have selected 9 acidic amino acid residue. Let me again note it down. How many acidic amino acid residues are there? There are 9 acidic amino acid residues. Okay, so what we have done, we have first selected and figured out how many basic amino acid residues are there. We end up identifying 18. Then we have selected how many basic acidic amino acid residues are there, and we found out that there are 9 acidic amino acid residues. Okay, so now based on these numbers, based on this data, the number of basic amino acid and number of acidic amino acid residues, now after comparing the number of acidic and basic residues in your protein lysozyme, would it be possible for you to expect the isoelectric pH or pi of this protein, okay, to be present above or below pH 7? Okay, so based on this data, number of basic amino acid and acidic amino acid, would it be possible for you to detect if the pH or pi of this particular lysozyme would fall below pH 7 or would it fall above pH 7? Okay, yes, of course you are thinking it right. This is of course basic amino acid residue, as its pH is known to be about 10. Why? You can clearly identify the, or you can clearly predict the isoelectric point or pi of the given enzyme just on basis of how many acidic or basic amino acid residues are distributed in your sequence, right? Well, in case of our lysozyme, this is a basic protein. Why? Because the basic side chains clearly outnumber the acidic side chains. Okay, the number of basic side chains, the basic amino acid residues, are double as compared to the acidic amino acid residues, and hence the pH of this protein is on the basic side, you can say that is around 10. Okay, so in this way, just by selecting and knowing the what type of amino acid residues, you can learn important properties of your protein. Okay, so now let us put away this layer info button, layer info windows, by clicking this close icon over here. Okay, so now let us see another way of selecting the amino acid residues. There are a few more tricks that are left to tell you. Okay, so what we would do now is that we would select only the you know select only the groups, or you can say we would select only the inhibitor group by selecting, by simply dragging these three amino acid residues. And if I press return, okay, I would then, what I have done, I have selected these three amino acid residue and hit enter. Okay, after hitting enter and after pushing the zoom center button, now we are only looking at the inhibitor of our protein molecule. Okay, now you should what you are seeing is a trineg only. So now let us kind of rotate it and get to know this trisaccharide structure. Okay, and let us kind of label all of the three groups by using this heading label. Okay, now you have labeled the three of those. So this is there exists a very powerful selection tool, well, that lies at the top of your graphics window. We are talking about this button. Okay, this button. Okay, eighth button in the bottom left, showing an eye and a circle. Okay, this button. So now if you click this button, okay, in a message space beneath it, okay, you can see that this deep view is telling us to pick one atom. Okay, or you can hit escape on your keyboard. Okay, so this is where your SPDBV kind of communicates with you. Okay, so this message space that is just present beneath the movement icon. Okay, deep view deep view is now instructing you to pick an atom. Okay, now let us click the nitrogen atom, which is present in a blue color on the center ring neg 202. Okay, it is not quite visible. Okay, if you again press escape, you would come out of that function. Okay, now let us recenter our molecule so that we can see the atom that we are supposed to click on. Okay, what we did is that we have selected this particular button. Okay, and now our SPDBV is asking us to select any or pick any atom. So what we are selecting is the nitrogen atom from our trineg inhibitor. Okay, if you click over here, sorry, okay, it is instructing us to pick. After clicking on this nitrogen, be sure that you select on the atom, not on the bond that connect the two atoms as such. you are supposed to click on the atom. Okay, so now after clicking, this dialog box has appeared. Okay, so now click to darken the first button, that is we would add to view the groups that are present within, we would type number 8 over here in the box, and after we click OK, see what happens. Okay, so what we have done, you have added to the display all the groups having at least one atom within the 8 angstrom of the atom you have picked up. So what we have done, we have picked up this nitrogen atom from this nag 202. Okay, and using this particular functionality, what we have done is that we have added whatever residues that has got at least one atom in the vicinity of 8 angstrom of this nitrogen atom. Okay, now let us now try to see ki how many amino acid residue have at least one atom that is present in the vicinity of 8 angstrom around this nitrogen atom. So let us try to label. Okay, let us try to select. Let us try to label, select it first. Okay, select visible group. Okay, now you can see that we have only added to the display the amino acid residue that has got at least one atom around the around your selected atom. It has been only selected, it has been only displayed, it is not selected. Okay, now let us select what is displayed. For that we would go to select and then select whatever groups that are visible. Okay, let us select whatever groups that are visible, select visible groups. And now if we click on this heading label, we would now see that in the, this is what you are supposed to infer. We have selected this nitrogen atom and there are how many amino acid residue? 1, 2, 3, 4. 4 amino acid residues that has at least one atom in the 8 angstrom vicinity of this, the selected atom from the trinag residue acid, that is this nitrogen we are talking about. Okay, so in this way, using this button, what you can do is that you can select the number of groups that are present in vicinity of a particular atom. That is what this button would enable you. That is how this selection would enable you. Okay, so uh, this is what it is done. So you know, more commonly you might want to display all the groups within a specific distance of the selected group. Okay, well over here I have shown you as to how many groups are present in vicinity of, you know, vicinity of this particular atom, this particular nitrogen atom, right? But most of the time you would be interested in finding out as to how many residues are present around some specific residues. Okay, so let us try to do these things in a different way now. Okay, so now first let us try to select and display only the trinag inhibitor, and now our objective would be as to see how many amino acid residues are present in 6 angstrom vicinity of this inhibitor. Okay, how many amino acid residues are present in the 6 angstrom vicinity of this inhibitor and not just this nitrogen atom. Here is the difference. Okay, so now we have selected this trinag and we would go to select and then we would select this neighbors of selected amino acid residue. Okay, and we want to display how many amino acid residues that are present in vicinity of 6 angstrom of your trinag. Okay, so either you can add to the view, or you can display the groups that are within, or you can select the group that are present in within. You can have different type of selection over here. Okay, type 6 in the box and you can click OK. After doing this, now you can see that you have added to the display all the groups that have at least one atom within the 6 angstrom of any known atom in your trinag. Okay, any known atom in your trinag inhibitor. Okay, so well, this is another way around, or this is another trick of selecting as to how many residues are surrounding any particular residues. Okay, so now there is one more trick that I would like to show you. Okay, so what you are supposed to do is that you are supposed to select and display and center the whole model. You would select the whole model, display the whole model, and center the whole model. Okay, means to select, display and center the whole model with side chains. Let us show all side chains over here, hide all labels, we don't need them. Okay, and you would simply go to display and show hydrogen bonds. Okay, so you do not have to compute hydrogen bonds every time. We have computed it last time and if you remembers them. So all you need to do is to go to this display and then show hydrogen bonds. Okay, so it will show all hydrogen bonds over here. Okay, so in case if you have restarted the computer program, then you might want to again go to tools and first compute the hydrogen bonds and then display it. Okay, so okay, now what we would do is that we would create a view in which we would only select this trinag. Okay, we have selected this trinag. Well, let us not remove other groups from the display. Okay, then we would go to this display and we would show only hydrogen bonds from selection. We would select this, show only hydrogen bond from selection. Okay, now all hydrogen bonds go away and whatever you have selected, only the hydrogen bonds that originate from the selected groups are shown over here. Do you note that? Okay, now again I would go to display and show only groups with visible hydrogen bonds. Okay, now it would show only the groups that are hydrogen bonded with the selected groups as such. Okay, with this simple set of command, that is display show hydrogen bond from selection and show only groups with visible hydrogen bond, using only these two simple commands, you have limited the display to what? To only trinag inhibitor and the residue to which it is hydrogen bonded. Let us zoom center it. Right. So what is on display? Only the trinag inhibitor and the residue which is hydrogen bonded to it. Okay, now what we would do is that we would select the visible group. Now you might note that although these groups are visible, okay, the hydrogen bonded groups that are visible, but they are not selected. Well, let us select all the visible groups. So now this becomes red. And I would show you one more trick. What we would do is that we would again go to select and use this inverse selection. Where is this inverse selection? Yes, over here. Inverse selection menu. Okay, now what have you noted? Okay, okay, and after that I would click on the heading ribbon and then I would zoom center it. Now a very interesting view might have appeared in front of you. Okay, now what you are saying is that you are showing your lysozyme model in which the trinag and its hydrogen bonded group are shown in wireframe model, the lines model you can see, with the rest of the model that is shown in ribbon. Okay, and you have learned one more thing, that the command select and inverse selection is handy way to select only the groups not currently selected. Okay, first we have selected these groups and then we have selected inverse selection. So this inverse selection would select all the groups that are not selected. Okay, so in this way, this inverse selection is handy way to select only the groups that are currently selected. Okay, that are not currently selected. So anytime you want to carry out operation on everything except the current selection, you should use this inverse selection command. Okay, so what do you see over here? You might see such representation in your biochemistry books, wherein the protein is usually shown in in cartoon representation, and the amino acid residues from your active site are shown in, say, some wireframe model, and inhibitor is also shown in some wireframe model with hydrogen bonding shown. Well, this is how you can create beautiful representation to show what you have found out in your findings, in your project reports, or in your assignments. Okay, so this is all I want to show. What we have learned today in today's demonstration section, we have learned as to what are the different ways you can visualize your protein molecule, what are the different ways you can select and display your protein molecules, and we have visualized our protein molecule in more efficient way in order to learn something from it. Okay, if that is the thing that you have learned, the objective of today's practical is over. Okay, so in our next session, we would be learning about measuring and coloring, all those stuff. Okay, so thank you for your kind attention. See you guys in our next session.